Cool. Well, hello. I'm Sandra Winters, and today I'll be talking about a method we've been developing for using touchscreens with birds. Many animals have striking appearances that are thought to protect them from predators. And there are a lot of different ways animals can do this, but I think some of the most interesting are ones where animals confuse predators while they're in motion. And some examples of this include things like flash coloration, where an animal displays conspicuous colors and then hides them. And this can kind of mask their movement trajectory or interfere with predator search images. There are flight patterns that can interact with the signal to increase efficacy of, for instance, warning colors, and some kinds of stripes, for instance, that can disrupt motion perception in predators. And an important way that we study how predators can drive the evolution of prey appearances is through experiments documenting how predators respond to prey. But this is particularly challenging in moving animals to get a stimulus animal that moves in a really characteristic way reliably. So a lot of these ideas, some of which are really old, haven't really been experimentally tested very well. So there are a lot of different experimental approaches for understanding predator responses to different prey appearances. These include things like predation experiments, where you chuck a bunch of models out into the field and see which ones get eaten. Behavior assays, where you have some sort of predator and show it something, maybe a moving robot. And then finally, particularly recently, it's been very common to do these kind of human clicking studies, where you get a bunch of participants to come in and click and serve as surrogate predators and click on prey. Of course, there's a kind of trade-off here between ecological validity and experimental control. And particularly in these human experiments, it's fantastic because you can get nice moving things on that screen, but that's quite divorced from the ecological context. So what we've been doing is trying to kind of unify these latter two approaches, where we can get natural predators playing predation games on touch screens. So that's what I'll just be showing today. So to do this, we're using a touchscreen equipped operant chamber, which is just an opaque box with a touchscreen in it. So it has, I'll just put all of these up. Things like viewing windows and access panels for ease of experimentation. And then inside, there's a display screen that can display any kind of digital stimulus. And then because some birds that can't flutter very well struggle to access a full screen, we have these assistive structures in front of the screen, which you'll see in a minute, that allow them to kind of hop around. And then the really key component here is this infrared frame, and this goes around the screen. And this is because bird beaks don't conduct electricity, so they can't use capacitive touchscreens like your phone. And so this is just a frame that projects infrared inside and can detect anything that pierces that plane as a click. Finally, we have a reward system, and we've tried a few, but we're currently using this magic box is what we're calling it, which is basically just a box full of good, tasty stuff that can be remotely controlled to open and close. My PI is super, super proud of developing that. So the display is the really key component here, and there are these layers. So there's the screen. We have a plexiglass protector because birds are destructive and messy. And then there's this IR frame, and then potentially at least these assistive hopping structures in the front. So these are a few images of one of our talk boxes. So you can see that there's the box controlled by a computer. Inside is a bird waiting to participate in an experiment. This is an old version where we had this wire lattice to help them hop around, but we've updated that more recently to use just horizontal acrylic rods. And we've had so many awesome interns. So this is Anna conducting an experiment. So for the experiments, we use wild-caught birds, which is really great because these are natural predators that haven't been raised in a lab, so they have the full range of experience. And we catch them in traps at Lammi Biological Station in Finland, and then we bring them into our lab there for usually a week to 2 weeks, bring them, release them back into the wild. So for our experiments, we've been primarily using blue tits, and this is because they're common in Finland, they're easy to handle, and these are a model system in visual ecology, so we know a lot about how a blue tit sees the world. So briefly, blue tits have very good color vision. They have 4 sensors in their eyes, so they can detect primarily red, green, blue, and UV colors. They have relatively poor spatial resolution. So at the top there, you can see how a blue tit would see that caterpillar at 10 centimeters, but even at the bottom there at 1 meter, it's quite blurred out. But they have very high temporal resolution. So a blue tit can see motion much better than we can. They see the world at about 131 frames per second. So that's much faster than us, much faster than your fancy screen at home probably. And of course, we can also use other types of birds, or at least small, manageable birds, but the blue tits have been working out pretty well for us. So this visual system information is critical for selecting the screen within this talk apparatus, because we want a screen that can accurately display as many colors as possible, so it has a wide color gamut. And then particularly critically, if you want anything that's moving, the screen needs to be able to play very fast videos or animations, which is beyond the capacity of a lot of screens. And I've actually spent a lot of time arguing with University of Helsinki and explaining, yes, I really do need this really fancy gaming monitor. Yes, it's actually for birds. Lots of fighting, but they let us buy it eventually. So probably some of you are thinking that these birds can see UV, and computer monitors usually don't display UV, and that's true. That's a key caveat at the moment, and we're working around that by just not displaying anything that would reflect UV on the system. But in the future, it would be really fantastic if we could get some sort of screen that can display UV and really fast-moving animations. So we'll see. All right, so here is an example of a bird playing one of our games. This is an early game. And the bird has been trained to peck the moth, and it's been trained that it has to peck the moth once and then catch it again, and then at that second peck is where it gets its reward. So, that's playing. So, it pecks this first moth that was easy to find. That triggers the moth to fly. Now the bird knows it needs to find it again. It goes to the wrong place and kind of missed out at first, but now it found the moth, pecked it. When it's pecked, it's released. You didn't realize that this audio would be on, but you can hear a little bit of blue tit sounds there. So it pecked the moth, and that peck triggered the screen to go gray, and then the bird could go get its food reward. And once they're trained to do this, they will happily participate in many trials as long as they're still a little bit hungry. Actually, some birds get really obsessed and they don't even take a food reward, and they just want to kill all the things. So individual variation. The major advantage of this approach is that you can run these interactive experiments, right? Where where the game responds to what the bird is doing. You're not just playing some sort of video that's on a loop, completely agnostic to what's actually happening by that predator. And of course, then you can have very controlled movement that's not based on, say, an animal behaving the way you want it to. And then you can get really accurate reaction times and peck accuracies as data. So there are a lot of things to consider when running experiments like this, and I'm not going to have time to go through many, but I put a list of some things to consider here, and I'll just highlight a few. One is validate your tech. If your screen starts playing your animations at 60 frames a second, it'll all look great to you, but the bird will see it as jumpy, at least if it's one of these species that has higher frame rate in their visual system. So it's really important to keep validating that, make sure that the monitor didn't get turned off and reset, oops, and you didn't notice. We've had that happen. Designing these experiments should be done very carefully, and there's a lot of things to consider. Reaction times require a start and a stop. And as much as these IR frames are really cool, they also detect anything that goes through them. That includes the bird's tail, for instance, so we get tail clicks, we get other types of clicks, so that's something that needs to be managed in the experimental design. The training stage is also really critically important, obviously, and it's usually by far the most time consuming. So we use the scaffolding approach where we first train the bird to like peck a little paper stimulus, and then eventually they get up to digital training and pecking, moving things on a screen. But it takes a while, depending on the bird and the project, of course. Usually it takes like 2 days to a week per bird to get them trained. And this can be automated. It's possible to automate it, but we've found that training especially is quite sensitive at certain phases, so it's better to do it manually so that you can reward like good tries that the computer wouldn't be able to detect. Of course, all of the design of experiments and everything should be based on the behavior and visual system of the species and the research question. So just to highlight, I think this touchscreen chamber is a great way to study moving anything, in this case prey. There are some drawbacks. Like I said, there's no UV on these screens, at least at the moment. The screen is 2D. You can get some 3D cues on there, but it's not the same as having a full like search arena. Of course, these tasks are a bit artificial, or very artificial. Birds don't play video games out in the wild, and it can be quite time-consuming to collect enough data for a full experimental design. But these drawbacks are balanced, I think, more than balanced by some advantages, which are that, again, you can get these interactive experiments with natural predators using really controlled stimuli that can move in controlled ways. You can get really precise reaction times and peck locations. And ultimately, I think this is a great methodological link between some of the experimental designs that are already being used. So it can combine really well with, for instance, something like a predation experiment. So just very quickly, I will give a shout out to George Hancock, who's a postdoc working on making some really cool animations for this system. And if you were a bird, wouldn't you want to play with one of those, right? And then if you're intrigued by some of these protective coloration and motion questions, stay tuned. My first year PhD student, Theo, is focusing on this for his dissertation. So hopefully there'll be some good stuff coming out in the coming years. So I'll just wrap up by saying that we've been using talks to study how avian predators influence the evolution of prey color patterns, but obviously this system can be used for a wide variety of other types of questions. Anything that you could test with a bird on a screen, and in fact, the technology would work with other species as well with the right types of modifications. So I'm really excited to see where this type of approach takes us in the coming years. Right, I think maybe I have a minute for questions. Well, I should remember to thank the many people involved in this experiment, and in particular, Ossie and Sunny, who put in a lot of work getting the system off the ground a few years ago. So with that, if there's almost no time for questions, maybe a very quick one. All right, thank you.
Okay, hi. My name's Ann Chambers. There we go. I'm a postdoc at the University of California, Berkeley, and I'm excited to talk to you about some of the work I've been doing on missing data imputation as it relates to genotype environment association methods. The main thing I've been working on is the California Conservation Genomics Project, and for those of you not familiar, the CCGP is a statewide initiative to obtain whole genome sequencing data from about 250 species across the state. The main goal here is really for us to understand how we can best conserve California's wildlife in sort of a multi-species context using genomic data. And one of the main questions we might have for each of the species in the project is sort of the genetic basis that underlies local adaptation, or being able to quantify adaptive variation. And one of the main ways in which we do this in the field is using these genotype environment association methods. And what these methods are doing is detecting significant associations between allele frequency declines and sort of a gradient of environmental conditions. We can input these environmental variables to understand what sort of significant SNPs, you can see those here in this Manhattan plot in orange, are significantly associated with those environmental variables. The issue with some of these methods is that they don't accept missing data. And so as researchers, we have a couple choices here. The first is that we can severely prune our data to the point where no more missingness exists in our data set. We run the risk, obviously, of missing true positives in our data set that we've pruned out, and we reduce power for these methods to detect our outliers. And so the other option, which is most commonly done, is what's called imputation, which is the estimation of missing genotypes using existing data that we do have. And that's what we're going to get into today. First, I want to show you some results from a few of our anonymized CCGP data sets. I'm going to show you some stuff for a bird, a montane mammal, and an insect. And each of these data sets, as one would expect given any empirical data, they differ in the amounts and degree of population structure, missingness, the patterns of missingness in the data sets, and of course the sampling that we see across the landscape. And so when we were beginning to think about adaptive variation and running these GEA methods, we wanted to test out a few imputation methods just to see how they would differ in these data sets. And so what we end up seeing is something like this. I've color-coded 3 different imputation methods using these colored boxes, also indicated on the x-axis, with the y-axis being just the number of significant outliers we detect from, in this case, redundancy analysis, or RDA. And what you can see is that there is variation in the number of these significant outliers we get, depending on the imputation method of choice. And what I'd like to emphasize here with these data sets is that they have very little missing data. We've really stringently filtered for missingness here. And at most, our insect data set has 2.6% sort of matrix-wide missingness. So this raises the question of what is going on with imputation that's resulting in us getting these different amounts of outliers. I want to take a step back momentarily and just walk you through how we impute missingness or impute values. And so what you can see here is an example genome with 5 individuals denoted by these sort of separated by dashed lines. And if you imagine the genome is a mosaic of information, we're trying to get at certain parts of it for quantifying this type of adaptive variation, you might imagine that this sort of red zone is what we really want to get at. These are our SNPs that are, serving, maybe they're climate-associated SNPs or serving some sort of adaptive function here. But we have missingness in a single individual. What we can do, and this is by far the most common approach, is median-based imputation. So we take the most common value, in this case across all 5 individuals, it's a blue genotype, and we assign that value to our missing individual here. Alternatively, what we could do is a slightly more sophisticated approach where we might want to impute based on our knowledge of population structure in the data set. In this case, maybe 2 of our individuals belong to the same cluster, and so we look at the individual that has a present site, which is red, and we assign that missing genotype according to that. There are additionally more sophisticated methods that exist, like imputing based on random forest or using phasing and using those phased haplotypes to infer missing, or to infer how to impute our missing values. And so we're going to explore 3 of those now. So in general, how does imputation affect the detection of our adaptive loci? And really, are we missing a signal of adaptation based on the imputation method that we choose? More specifically, for different imputation methods, I'm going to ask how the amount of missingness impacts the detection of adaptive loci. This child is really upset. Just making direct eye contact. Okay, so to do so, we're going to start with 2 simulated data sets. The first of which is more reduced representation type. The second is more whole genome sequencing type, and I'll get at that in just a moment. Then we're going to selectively remove sites and vary the total proportions of missingness in each of these data sets, ranging from 1% missing to 50% missing, which we might see in those reduced representation data sets. We're going to then impute missing values using median, random forest, and population structure-based methods. For structure-based methods, we're going to vary k from 2 to 10. And then we're going to run redundancy analysis as our GA method and look at our results. I wanted to take this second to plug Alligator, which is a package that we developed recently in our group. This is a package to do a number of landscape genomics methods, and included in there are a couple of these GEA methods. And that's the way I analyzed these data and imputed missing values. For those simulated data sets, our reduced representation type data set, this is an empirically parameterized data set that was produced by Ahrens et al. In 2021. And it's parameterized using this eucalyptus data set of 26 populations. So this is a population-based data set with about 24,000 SNPs, so maybe more representative of your sort of typical RAD-seq sized data set. We did also want an individual-based, spatially explicit simulated data set, and so we used the program Geonomics. This is a highly flexible sort of simulation platform, and it's kind of neat because we can actually control that adaptive surface upon which individuals evolve. And this data set size was a bit closer to our whole genome data sets, but still sort of manageable, of 100,000 SNPs. And across this landscape, we subsampled 150 individuals to mimic sort of typical individual-based sampling. And we also ensured, we ensured that there was some population structure that was sort of biologically realistic as best we could. This was difficult to do, surprisingly. Okay, so in terms of our results, first I'll show you our reduced representation type data set, that Ahrens et al. Data set. This is the minimum amount of missing data. At 10%, you see there's not much of a difference between our imputation methods. And again, this is color-coded consistently across all of our plots. As we increase those proportions of missing data, we see an increase in the number of total outliers detected. And you can see that dashed line here is the number of adaptive loci we know exist in our simulated data set. And so most strikingly, it's really all about the amount of missingness, i.e. the amount of imputation that we're doing that dictates this increase in significant outliers found. But there does seem to be also an increase in outliers we find as we start subdividing the data up into more and more of those population clusters, as we start increasing K, as you can see. But what about what these outliers actually are? Just because we have a number that might be close to those true number of adaptive loci, how many of these are true or false positives? And we can measure the proportions of these using the true positive rate and a false detection rate. And so what we see in this data set is a gradual decrease in our true positive rate that we want to maximize as we start subdividing those data. And oppositely, we see an increase in false detection rate as we increase that K cluster. So these data kind of indicate to us that median-based imputation is performing actually quite well, which is really great news for me, for those of us who do these GEA methods, because it's very quick and simple to do. When we look at our WGS data set, however, we see kind of a different story. So I want to remind you that, I don't think I mentioned this, the Ahrens data set had essentially no population structure. And our WGS type data set we simulated, we purposely really wanted to ensure there was some population structure there. And so what we see is that we see quite, more drastic changes within imputation methods to the number of outliers that we detect. I also want to highlight here that the y-axis is very different. There are only 6 adaptive loci in this data set, so that's also why we see very different jumps between these data points. Once again, if we look at those true and false positives here, we can see, interestingly, that as we increase the K values from 2 to 10, we do see potentially a reduction in our false discovery rate and an increase in our true positive rate in this data set that does have population structure in it. I also want to emphasize here, this is results from one simulation, so we intend on performing many more of these and getting some idea of if this is a consistent pattern among our simulations or not. Okay, so sort of to make some general conclusions here with these preliminary data, we can see that the amount of imputation required really is the most important consideration in detecting our outliers. I think this is quite intuitive for all of us who have worked with these types of data. We really want to try to be quite stringent in how we filter out missingness. I was quite surprised that median and random forest-based imputation performed quite similarly, not just in the number of outliers detected, but also what those outliers were. I say this is surprising just because the computational requirements between these two methods are so different, and the sophistication behind, the statistical framework upon which they work is also very different. But that's, I think an interesting finding worth investigating further. And lastly, potentially in data sets with population structure, there may be a benefit to structure-based imputation in obtaining more true positives and correspondingly fewer false positives. But we need to investigate this a little bit more with running our simulations many more times. I want to take the last minute or so to talk about some future directions of this project that I think are quite interesting. If we go back to two of our CCGP data sets from our insect and our bird, I was really intrigued initially by not only the amounts of missingness in our data sets, but the patterns of missingness. And so I made just some neighbor joining trees, and I'm showing you here the tips of this tree are colorized based on individual-based missing data proportions. And what you can see based on these lambda values on the bottom, there's a very strong phylogenetic signal to missingness. And the trees themselves I reconstructed as well with no missing values, so it's not that the missing data is driving the topology of these trees. And this is also in line with the population structure that we see in these data sets. And I think there's many hypotheses we can make for why similarly related individuals would have similar proportions of missingness. Certainly in RADseq data sets, I think this has been shown many times. But this leads me to ask kind of an extended question to this project, which is how do patterns of missingness impact the detection of our adaptive loci? Especially in our CCGP data sets, the vast majority of ones we've looked at have this signal, and a quite strong one. And so I think it's really worth delving into this. So I'm right now working on selectively removing our sites in a non-random way, which is kind of an interesting problem, and I'd love to chat with anyone who has ideas for how to do this at any point. Okay, so I'm going to end there. I want to really want to thank Colin Ahrens for helping me out with his data set. He was extremely helpful and willing to, and excited by the project, which was great. I want to thank Drew Terasaki-Hart, who wrote Geonomics, and I encourage you to take a look at that software. It's really wonderful. Anusha Bishop in the Wong Lab. Eric Embody at UCSC has been our bioinformatics wrangler for the CCGP. He's an incredible biologist. Members of the Wong Lab and our CCGP core team. And if you're interested in checking out the CCGP or reaching out to me, the info's here. Thank you so much, and I'll take any questions.
Hi, everyone. My name is Rob Porch. I'm a PhD student in the University of Oregon, and today I'm going to be talking about sort of two interrelated projects that I've been working on for the past couple of years during my PhD. One of which is building a large phenotypic database on the metabolic traits of prokaryotic species. And the next one is using this database as a central tool to write a software that will help predict the phenotypic composition of microbial communities that are 16S based. So a little bit of background, if you don't really think about prokaryotes that much. They're overwhelmingly abundant and diverse and the most ancient life forms on the planet. Whenever I talk about how cool prokaryotes are, I like to show this phylogenetic tree, which sort of puts eukaryotes in their place. They're just that tiny green sliver at the bottom, and the rest of the tree of life are prokaryotic organisms. To date, we've named roughly 40 bacterial phyla, but it's estimated with genetic evidence that that number could be upwards of 1,300. So we're really just still discovering just the sheer diversity of prokaryotes, and with that diversity, they're able to occupy incredible niches across many different environments, even really extreme environments that eukaryotes couldn't even dream of inhabiting, like really extreme hot springs of high temperatures. And fortunately for microbiologists, over recent decades, there's been a rapid accumulation of sequencing technology that has allowed us to understand natural prokaryotic communities better. So in the form of like marker gene sequencing, like targeting the 16S gene, which is very slow evolving in prokaryotes, we're able to understand the taxonomic profile of prokaryotic communities better. And with metagenomics, we can not only see the taxonomic variation and taxonomic data in microbial communities, but also genetic data, such that we can infer the function of those microbial communities based on their gene content. But what I want to point out here is I've also plotted the number of cultures done in labs. And what's really lagging behind now is direct empirical evidence of phenotypes of prokaryotic species. So we can sort of think of this, the distance between these two curves, as the lack of phenotypic data that we have for prokaryotic species. A lot of the prokaryotic species now that we have genetic data for have not yet been cultured, and sometimes microbiologists sort of dramatically refer to them as microbial dark matter. And this is sort of a problem, especially if you're a microbial ecologist, because in ecology, we like to think about communities not just in terms of their taxonomic composition, but their functional composition. So not just who is present in the community, but what it is they're actually doing in their community. So bridging this gap between prokaryotic taxonomy and function is a really tricky sort of problem that we're left with right now in the field. And to bridge that gap, we sort of have to rely right now on computational methods. And this is where my two sort of projects come in. So I'm going to be talking about both. The first project, like I mentioned at the beginning, is building a phenotypic database of prokaryotic species that have been cultured. And then the second project is developing a software called FabriTax2, which stands for Functional Annotation of Prokaryotic Taxa, which is a software that will predict the likely phenotypes of organisms detected in 16S-based microbiome surveys. So a little bit going into the methods, so what traits we've considered for the database, sort of the workflow of how we compile the phenotypic data for this database, as well as the broad functionality of the software. So first, the FabriTax2 database focuses primarily on the metabolic capacity of prokaryotes. So when we think about like the phenotypes of prokaryotes, a lot of the time we like to think about their metabolism because it's really important for how they interact with each other in their environment. And this is just sort of to show the breadth and the depth of the traits that we're including in the database. So we have traits related to chemoheterotrophy, phototrophy, many different forms of respiration and fermentation, nitrogen cycling, methanogenesis, and some more traits that I was not able to show here. And then there's also a depth in those traits as well, where they sort of have a nestedness in specificity. So how do we compile the data for the presence and absence of all of these traits? So the data are collected manually at the species and trait level, and part of this is working with a wonderful and amazing team of undergraduate researchers who I sort of have to get them really excited about microbial metabolism, which is a huge feat in itself. It's a huge literature search project, essentially, where we're looking for empirical evidence for the presence and absence of all of these traits in the database. So really quickly, I just wanted to demonstrate sort of like the logic that we use and the workflow that we use to compile these data. So this species, Thermoanorobacter ethanolicus, just as an example, we go through the primary literature, so papers published in journals such as IGSEM, as well as the Bergey's manual, which has very conveniently formatted phenotypic data of cultured prokaryotes. So we start at the genus level and see if there are any like broad sweeping traits at the genus level that all species within that genus share in common. And we look for like direct evidence of the presence or absence of these traits. But sometimes we actually have to deduce the presence or absence of traits. So an example of that here is the fact that members of this genus are obligate anaerobes. So what we can infer based on that information is that they are absent for aerobic metabolic traits, such as aerobic ammonia oxidation and nitrification and so on. And then once we finished going through genus level information, we then go to the species level and see if there are any more traits at the species level. And here, based on the literature, we can see that this organism is present for fermentation, more specifically alcohol fermentation, which you might have guessed based on the name of the species, gelatin degradation, and is negative for cellulolysis. So switching gears to the software, like I mentioned before, this phenotypic database is going to be a huge part of the FabriTax2 software, and this is just a diagram showing the functionality of the software. So broadly speaking, the input would be the user's microbial community data in the form of 16S sequences. So that could be either a list of the OTUs in their community or a relative abundance table of OTUs in their community. And the software will match the OTUs from the user to records in the phenotypic database. And for any OTU that is not already represented in the database and the reference phylogeny, we'll use a phylogenetic placement tool to place them onto the phylogeny. And then the software employs what's called hidden state prediction algorithms to compute the likelihood of those unrepresented tips being present or absent for all of the phenotypic traits in the database. Really briefly, I just wanted to mention how we build the reference phylogeny. So we match the species records in FabriTax2 database to Silva records, and we use the full read 16S sequences of those Silva accessions to build this phylogenetic tree using a GTDB backbone. And that's how we get this reference phylogeny. And then the output of the software, instead of taxonomic data, is predicted functional data. So again, going from who is present in the community to what are these community members likely doing. And if the user's input was a relative abundance table of OTUs, the software will print out a relative abundance table of phenotypes. So really quickly, I wanted to elaborate a bit more on the HSP algorithms that we're using for the software. So the first one is maximum parsimony, which is not model-based and just tries to minimize the number of trait state changes in the tree. And then the next two HSP algorithms are model-based. The first one uses a continuous time Markov model, and the second one uses a binomial distribution model. And the really cool thing about these is that they're likelihood-based, so from the user's end, they can adjust the threshold at which those traits are predicted to be present. They might care more about minimizing false positives versus false negatives, for example. So in terms of the phenotypic database, this is just a summary of all of the taxa for which we have data so far. So we have phenotypic data for about 10,000 species to date across around 36 phyla. I'm going to go through this a little bit quickly, but what we can see here, this is not new to us, but we see that there's a huge taxonomic conservation for some of these traits, like oxygenic photoautotrophy, methanogenesis, et cetera. So this sort of just tells us that we've accounted for a lot of prokaryotic species on the phylogenetic tree of prokaryotes. So looking at the accuracy of the HSP algorithms, on the x-axis are the algorithms, and the y-axis is the balanced accuracy. This is the average between the true negatives and true positives using leave-one-out cross-validation. Each point is one of the traits in the database. The size of the point is the phylogenetic conservatism of the trait, and the color is the frequency of the trait, so the fraction of tips that are present for that trait. What I wanted to show is that there's a loose correlation between conservatism and accuracy. So we can see that oxygenic photoautotrophy and methanogenesis are quite easy to predict, and they're the most conserved traits in the database. But even less conserved traits like chemoheterotrophy, for which we just have a lot of presence-absence data, are also quite easy to predict. This is the elephant in the room, are these traits that have a 50% balanced accuracy, no better than a coin flip. So these are more lifestyle traits, and I'll talk a little bit more about reporting bias, but it's very, very rare that the literature reports the absence of these traits, and I think that might have something to do with why these traits are so difficult to predict. This is just comparing, again, the accuracy of the HSP algorithms in pairwise combination, and this is just to show that by far maximum parsimony, if you look at the center and right panels, underperforms compared to binomial and MK model HSP algorithms. And on the left panel, we can compare for each trait how well the MK model and binomial HSP algorithms perform. And we can see that generally each trait is performed, like the accuracy is quite similar, but there are some outliers here. And the good thing is that we have options for HSP algorithms. So when we publish the software, we will choose a default HSP algorithm for each of these traits based on which HSP algorithm performed the best. Lastly, really quickly, I wanted to get into reporting bias. So there's this reporting bias in the literature where most often the presence of a trait is reported and not the absence. And what we do with those unknowns is a really interesting question and sort of like goes back to the previous talk. So we're left with the decision now, do we treat the unknowns as absent or do we just omit them entirely from the predictions? And this is looking at the true positive rates under each of those scenarios in the leave one out cross-validation. And the closer they follow this y equals x trend, the less influence there is of reporting bias in reporting the presence of those traits. So it's reassuring to see that they generally follow this trend, but there are some outliers where one scenario may provide higher accuracy in the trait prediction compared to the other. So the next steps for these projects is to prioritize trait level data collection. I think this might help with the traits that have shown to have like pretty low accuracy. So really prioritizing, including more tips into the reference phylogeny for which we know those tips are present for those traits. I'm also looking to recruit some early users of the FabriTax2 software. So if you think about microbial communities, if you have 16S data, I'm really happy to chat with you about that. I would love some useful feedback and suggestions on what we can include in the software. And then some further projects that might be really interesting is to incorporate genomic data and see if there are some genes that have been previously poorly understood that might actually be strongly linked with the presence of these phenotypes. So with that, I really want to thank the undergraduate team. They worked so hard with me learning microbial metabolism and doing literature search for this project, as well as my PhD committee. And I might have time for one quick question. Thank you. Yeah. Are you thinking about pie crust? Yeah, I can talk to you about the difference between the two. They have a similar philosophy, but they fulfill different niches. But yeah, I can explain it more to you if you're interested.